Good morning for you, good afternoon for those who are in Europe. I'm a PhD student from the University of Groningen in the Beth Bulmas group. And in our group, we mainly focus on membranes and membrane proteins. So in this work that I'm going to present now, we studied the biophysical properties of membranes that mimic those of the minimal cells of the JCVI-syn3A. So just a quick introduction. So we all know what the minimal cell is. So basically, where all non-essential genes are eliminated from bacteria while preserving their essential function. But while the metabolism and the genetic processes have gained a lot of attention, not much is known about the lipidome composition. And identifying a minimal viable lipidome is essential for understanding the lipid structures, the physical properties that support the membrane integrity and therefore function. So we know that both lipidome, those of mycoplasma and JCVI are dominated by cholesterol, which accounts for at least half of the total lipid abundance. And this is well known for mycoplasma, which lack a cell wall and possess only a single plasma membrane, where cholesterol is needed to strengthen the membrane function, the membrane function, and modulates properties such as fluidity and permeability. And on the other hand, we know from the interim book study that this is the composition for the minimal cell, and compared to the composition of its ancestor, so that of mycoplasma, the JCVI minimal cell has higher cholesterol content and lower PC content. And on the other hand, PG and cardiolipin, which are synthesized internally, are conserved, meaning that the regulation and the synthesis of these two lipids are also conserved in the minimal cell. And so this highlights an important feature, meaning namely that unlike the proteome, most of the lipidome of the JCVI is therefore not genetically encoded, but is shaped by environmental availability and biophysical constraints. And this highlights the membranes as a sort of an evolutionary constraint. So yeah, I was saying that so this highlights the membrane as an evolutionary constraint where genes can be reduced extensively, but the lipid composition cannot be arbitrarily altered without losing availability. So this implies that once optimal lipid set is established, it became conserved in a certain organism, providing not only stability and control permeability, but also a sort of universal physical framework for the persistence and origin of cellular life. So our work addresses a central question, that is, which membrane properties sustain minimal cells, and how the balance between condensing lipids such as cholesterol, sphingomyelin, and fluidizing lipids such as cardiolipin, POPC, and GPG defines the chemophysical identity of JCVI minimal cell. So we investigated the structure, the molecular packing, and the permeability of membranes and of LUVs, so large unilamellar vesicles made of the same composition of the JCVI, so that we call minimal cell 1 or mix 1, and we systematically changed the ratio between cholesterol and POPC, and this composition mimic more the membrane composition of the membrane of mycoplasma. And then we also tested 20% cholesterol and 44 mol% POPC. And so to begin, we asked whether vesicles could be formed at such high cholesterol content, since such high cholesterol content is quite unusual for the microorganism and also for us to make vesicles. But we saw with images that vesicles are formed robustly under all conditions, and showing both unilamellar and multilamellar vesicles without major defects. And when we measured the biolayer thickness across 37 vesicles, we found only a modest increase in the only modest increase in the thickness with a higher cholesterol. However, the experimental error is quite high, meaning that any cholesterol dependent effect on thickness appears limited. And we know in simple systems such as POPC or DMPC that cholesterol is known to produce a strong thickening effect, while here the overall impact of cholesterol appears to be minimal. And this can be explained either by a compensatory effect from POPC or by the fact that the condensing effect of cholesterol and sphingomyelin might already be saturated and reaching a plateau where further increase in cholesterol no longer translate into measurable thickening. And the condensing behavior of cholesterol is supported by Langmuir monolayer analysis, which provide a direct insight into the molecular into the packing properties of the lipids in the monolayer. Here we analyze the isotherm in with respect to the lift of area, that is the first value at which the molecules start to interact and the surface pressure is detected. So basically it represents this point in the isotherm when the isotherm start to take off. And a high lift of area indicates weaker interactions, weaker intermolecular interactions, or bulky structures that prevent tight packing. And here we can see that with the progressive replacement of cholesterol by PLPC, the lift of area increases, reflecting a looser packing and a gradual shift from a highly condensed cholesterol sphingomyelin dominated regime toward a more fluid state dominated by PLPC, cardiolipin, and DOPG. And the second parameter we calculated and I wanted to show here is the compression modulus. The compression modulus represents the peak here represents the most compressed state in the monolayer. Here we can see that it decreases with decreasing the cholesterol content, and this supports the idea that the membranes became more compressible and less ordered when less cholesterol is present. And also this value also provides information about the physical state of the membrane, and this value fall in a range between 100 to 250 millinewton per meter, meaning that these membranes are in the liquid condensed or liquid ordered state, and this is precisely the regime associated with the cholesterol sphingomyelin rich lipid rafts in biological membranes. And such rafts combine high acyl chain ordered with lateral fluidity and have been shown to be stable yet deformable to provide all the let's say the functional microdomains for protein recruitment and function. And so here from this study, from this results overall indicate that there is an interplay between fluidizing lipids such as cardiolipin, PLPC, and DPG, and condensing lipids such as cholesterol and sphingomyelin. And the interplay between this lipids keep the minimal cell membrane in a liquid ordered yet functionally flexible state. Then to probe the lipid miscibility, we measure the excess area and the free and the excess free Gibbs energy, when they are negative, it means that there are favorable interactions and the components are well mixed, while when they are positive, it means that there is a tendency two unfavorable interactions. And in this case, we observe that in the mix 1 and in the mix 2, so the ones with a 40 and 60% of cholesterol that are consistently negative, it means that there are strong attractive interactions, mainly driven by cholesterol and sphingomyelin association. But here what is important is that this doesn't mean that the monolayer is homogeneous. Instead, the cholesterol sphingomyelin rich domains coexist with more fluid regions enriched in POPC, cardiolipin, and DOPG. This just means that since cholesterol is known to be well mixed and more soluble in saturated lipids, this indicates that cholesterol and sphingomyelin stabilize the monolayer through ordered domains, and in the on the other hand, monolayers with lower cholesterol content tend to be slightly positive, and they have an excess area close to 0, indicating weaker interaction and looser packing, in agreement with the fact that the monolayer is more loosely packed and less ordered. And consistent with the Langmore budget analysis, the permeability measurement also showed that PLPC, cardiolipin, and DOPG counteract the strong condensing effect of cholesterol and sphingomyelin, preventing a shift toward a very low permeability, where transport would rely on specific transporters. And here we can see that as the cholesterol content increases from 20 to 60%, the permeability of water, ribose, and glycerol decreases, consistent with a transition from a more disordered and less hydrophobic membrane to a more compact and ordered and more cholesterol-rich membranes. So here, water remains the most permeable solid, while ribose and glycerol have a permeability still in a biological range, so in the order of 10 to the power of -8, -6. And this is particularly important, especially for glycerol, because this means that glycerol can still diffuse in and be part of the synthesis of PG and cardiolipin, and also for the virulence of certain pathogenic mycoplasmas. And so also these confirm that the minimal cell membranes combines ordered and for stability with just enough permeability to sustain life. So to conclude, we saw with cryo-TM that the vesicles and LUVs that mimic the composition of the minimal cell are quite robust, well formed. Some of them unilamellar, some of them multilamellar. We didn't observe any significant change in the bilayer thickness while replacing cholesterol with POPC. Overall, the cholesterol dries, packing, and ordered, but all the mixtures, either with 60, 40, and 20% of cholesterol, are in the liquid ordered state, so where there is order in the lipid chains, but still flexibility. The lipid mixing and the cholesterol and sphingomyelin stabilized from a thermodynamic point of view the monolayer and the membranes. And consistent with all of these, also the permeability tend to be affected by the presence of cholesterol, while increasing cholesterol decreases the permeability, but still being in a biological range. So overall, while cholesterol and sphingomyelin condense membranes, POPC, DOPG, and cardiolipin counteract to maintain fluidity and heterogeneity. And I want to acknowledge Bert Pullman, my supervisor, and also the other people who worked in this project, and also my group, the membrane enzymology here in Groningen. And this is it.
That's terrific, Caterina. Questions?
I was wondering if you had taken a look at the E. coli plasma, lipid composition and, how it manages to function effectively without cholesterol.
How do I manage to function without cholesterol? Could may I ask you to repeat the question, because I didn't fully get the question.
The E. coli plasma, mycoplasma, clade is able to function without any cholesterol, and I was wondering what the effect of that has on its membranes, and what might be determining its ability to do that.
Yeah, well, for sure, without any cholesterol, the membranes, overall, the whole physical state of the membrane will shift more toward a completely fluid state. So I guess that a membrane, I mean, it's something like this, but without cholesterol, would function more similarly to, for example, E. coli, that is a bacteria that doesn't have any cholesterol, but it has a membrane composition made of cardiolipin, DOPC, DOPG, DOPE. So I guess that from a biophysical point of view, the membrane will be more similar to that of E. coli, where the membrane is mainly in the fluid state, and for sure also the permeability will be faster. So also the kinetics of and the diffusion of the biological osmolalite will be faster, and so I assume that a mycoplasma without cholesterol will be more similar to E. coli, for example.
Thank you.